Good afternoon and welcome to today's Public Health Ontario Microbiology Rounds presentation on Oropouche virus, what you need to know. I'm Dr. Angela Ma. I'm a clinical microbiologist at Public Health Ontario, and I have the pleasure of moderating today's session. Firstly, I'd like to acknowledge that the land we are gathered on is a traditional land of many nations, including the Mississaugas of the Credit River, the Anishinaabe, the Chippewa, and the Haudenosaunee and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis people. Toronto is home to Indigenous peoples from across Turtle Island, and we are very grateful to be living and working on this land. In the spirit of peace, friendship, and respect, we are committed to building and maintaining relationships with our Indigenous partners. I am now pleased to introduce today's speaker, Dr. Sean Clark. Dr. Sean Clark is a clinical microbiologist at Public Health Ontario, where he oversees laboratory testing for genomics, arthropod-borne viruses, and viral hemorrhagic fevers. Without further ado, I'll let Dr. Clark take it away.
Good afternoon, everyone, and thank you, Dr. Ma, for the introduction. As well, thank you to all of those who have joined us on today's PHO Microbiology Rounds. I wanted to speak today about an emerging or perhaps re-emerging arbovirus that has been of broad interest for the past several months. And you may or may not have heard of this virus prior to today's presentation. However, at the end of today's presentation, I hope that you're able to take away some tidbits and useful information that you can use going forward. I have no commercial disclosures to declare. And in addition to my role as a clinical microbiologist at Public Health Ontario, I'm also an assistant professor in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. Now, today's presentation, there's 4 key learning objectives, and we'll explore each as we move through the hour. The overall objectives for today's talk on Oropouche virus are that you as the participants will be able to describe the important virologic properties of the Oropouche virus and related zoonotic and vector-borne viruses, to explain the current epidemiologic patterns of Oropouche virus infections in the Americas and how it may impact people living in Canada and specifically in Ontario, to discuss important drivers that may be contributing to the current Oropouche virus outbreaks, and finally, to describe laboratory testing methods and algorithms for the detection of Oropouche virus infection. And with our final learning objective, we will be looking at testing that is available through Public Health Ontario's laboratory. Now, it's possible that you may not have heard of this virus prior to today's presentation, let alone know how to pronounce it, because again, it's not something that's common to us here in Canada. But it was mentioned recently in a comment on another call by Dr. Aaron Staples from the CDC that depending on your language of origin, the name of this virus can be pronounced slightly differently. So depending on if you have French language origin, English language origin, Spanish language origin, it's possible there may be some slight differences in how you pronounce this virus's name. But for the purpose of today's presentation, I'll refer to it as Oropouche virus. Over the past several months, this virus has been a hot topic in both print and social media within Canada and beyond. And this is evidenced by some examples of news headlines that I've sourced from the end of the summer from CBC News and Global News here in Canada, as well as other news agencies. Together, these news agencies highlight that there's an ongoing concern regarding infections caused by a virus that not many may be familiar with. And along with these news headlines, even some of our furry friends have received a bit of a bad rap, with sloths having been implicated and highlighted in the transmission cycles of Oropouche fever and Oropouche virus disease. Now, this virus, and in specifically these news articles, has really generated some more widespread recognition about current and ongoing Oropouche virus outbreaks, not just among healthcare providers, but also among returned travelers themselves. Now, it's important to recognize that this is not something that is a brand new virus that has just sort of come onto our doorstep in 2024. The Oropouche virus was first detected in 1955 in a febrile forest worker in a village in Trinidad and Tobago that's referred to as Vega de Oropouche, which is located near the Oropouche River. So here we have an image of Trinidad and Tobago on a world map. The map highlights in black the region where the Oropouche virus was first discovered. And the South Oropouche River, which is highlighted here in blue, is the primary location where that febrile individual was first identified. Now, this river basin runs along the southwestern part of the country, and it is an area that is known to have significant swampland, wetlands, and vegetation, which together help to sustain the vectors that are known to transmit the Oropouche virus. And we'll talk about those a little bit later. But since its discovery in 1955, the virus has actually been identified as being endemic to the Amazon basin region further south in South America. And a significant number of case reports in the literature indicate that there's a wide geographic distribution across the region. So we've established the virus exists and that it's of concern to human health, primarily through reports from print and social media. But what are the key features of this virus and how does it compare to other related viruses? The formal name for this virus is orthobunyavirus oropouchense, or again, as we mentioned before, the more common name Oropouche virus, or you may see it abbreviated on some of my slides as OROV. It is an emerging arthropod-borne virus that is primarily a member of the Peribuniaviridae family of viruses. And within that family, it is a member of the genus Orthobunyavirus. These are viruses that are single-stranded, negative sense RNA viruses, and they're primarily known to be transmitted through arthropod vectors, and that's the main route of transmission to humans. It's known that these viruses, well, Oropouche in particular, circulates in South America and the Caribbean, and it's known to be endemic in many South American countries, and in particular, Brazil is known to be an area where there is a high number of cases annually. Outbreaks of Oropouche virus infection are not novel. Again, the virus itself is not novel, nor are outbreaks of the virus. And these have been known to occur in the past 10 years within the Amazon region, again in South America. And before the emergence of the chikungunya and Zika viruses, which are also arboviruses associated with human infections, it was claimed that the Oropouche virus was actually the second most frequent arbovirus infection in the Amazon region, and particularly Brazil, after dengue virus. However, our information on that is really limited, and the true burden of disease is likely not really well understood due to challenges with the detection and surveillance of this virus, which may not have existed previously in many of the affected regions. Now, the Oropouche virus has a similar structure to other members of the Peribuniaviridae family. This is a cartoon image of the virus and some of its viral genetic material. Over here on the left, we have a cartoon depiction of the virion, which is approximately 70 nanometers in diameter. And this is an enveloped virus, as we can see by the outer layer that's surrounded by glycoprotein spikes. The genome of this virus was first reported, and genome sequences from this virus was first reported in literature about 16 years ago. And since then, not really a lot extra has been known about the virus itself. What we do know is that the virus is comprised of 3 negative sense single-stranded RNAs. So this virus is classified as a segmented virus. Each of these segments is referred to by a specific an alphabetical letter. We have an S segment, we have an M segment, and we have an L segment. Each of these segments encode different proteins that are essential for virus replication and other parts of virus life cycle. The S protein encodes the viral RNA polymerase. The M protein encodes viral glycoproteins and non-structural proteins that are primarily involved in viral attachment and entry into host cells. And these in particular are important because these are the primary antigenic sites. And this M protein is under selective pressure, and we'll understand the reason for that a little bit later on in the talk. The S gene encodes the nucleocapsid and some other non-structural proteins. Now, the importance of this virus being tri-segmented is that this virus's genome is susceptible to reassortment. And what happens essentially there is that new viruses that are derived from Oropouche virus can be created following a transmission event within a host cell. And again, we'll talk about that in a few slides. But takeaways from this slide would be that the M segment and the S segment are important for the detection and characterization of Oropouche viruses in human infection. So we mentioned that this virus is part of the Orthobunya virus genus, but what does that sort of mean in the general scheme of things? Well, essentially it means that there's a lot of related viruses within this genera that also cause disease in humans and animals. And what we have here on this slide is a phylogenetic tree that's depicting the genetic relatedness of viruses that are classified under the Orthobunya virus genus. And this is based primarily on the sequence of that M gene, which is where the majority of variation in Orthobunya virus sequences originates. Each colored dot on this phylogenetic tree represents a different unique virus within the genus. And the length of each branch here, again, I've turned this phylogenetic tree on its side, but the length of each of these branches delineates the number of genetic differences between each of the viruses within this genus. What you can see essentially is that there's a large amount of diversity that is present, and there's around 50 virus species within this genus. Around 30 are known to affect humans and animals, and I've highlighted two areas in particular on this phylogenetic tree. On the left, we have what's referred to as the Simbu serogroup. This is a series of closely related Orthobunya viruses that contain Oropouche and Oropouche-like viruses. So we have Oropouche on the top here, and we have two of its reassortants, Iquitos virus and Perdoes virus. And these are all members of the Simbu serogroup. But for all intents and purposes of today's talk, Oropouche virus is going to be our main concern. Now, these viruses do not circulate in Canada, but what you may be familiar with is other Orthobunya viruses over here on the right-hand side, which are found to circulate in Canada. And these include the snowshoe hare virus, California encephalitis virus, as well as Jamestown Canyon virus. And these viruses all have that same tripartite genome and are also susceptible to reassortment. So understanding that the virus's genome is segmented and can create these reassortants, but what does that sort of mean in the general scheme of things? What it means is that the virus can produce multiple different variants of itself that can have differences in not only genetic material, but also ability to infect different vectors and its ability to be competent in different vectors to be competent for the sustaining this virus vector out in the natural environment. So this figure depicts a potential process for how segmented viruses, such as Oropouche virus, can undergo genetic reassortment. So this reassortment can occur when two viruses co-infect the same host cell, and the swapping of some genetic material can create novel variants of the original origin viruses. Here, what's anticipated is that the Oropouche virus can reassort with other unknown Orthobunya viruses, and they've created, again, these reassortants. So again, the Iquitos and Perdos virus that we mentioned before, as well as the Madre de Dios virus, which is another reassortant variant of Oropouche virus. These viruses are all of concern. However, Oropouche is the one that is currently responsible for the current outbreak. What these phylogenetic trees here are showing is that there is very little sequence diversity amongst the S and the L proteins and genes, but it is really the M segment, which is where the underlying variation comes from, and that is what's driving primarily the reassortment of these different viruses. So where is Oropouche virus found? How does the infection occur, and why is it really such a concern in 2024? Well, It's important to know that we really don't know much about the virus itself. And I think that's one of the important things that I'll be trying to highlight in today's presentation is there's a lot of knowledge that's lacking regarding this virus. The transmission cycle of the virus is really not well described. There are perceived host and vectors, but there's really much that remains unknown. So this describes a potential schematic that's been adapted by DeRosa and colleagues. And similar to other arboviruses, it's believed that there are two phases of the transmission cycle. There is a sylvatic phase and an urban phase. So the sylvatic cycle over here on the left um, is thought to involve several different vertebrate hosts. This primarily is also anticipated to occur in forested areas and can include vertebrate hosts such as a three-toed sloth, which again we saw implicated in some of those news headlines earlier on in the presentation. Some non-human primates and some rodents and organisms such as the marmoset, as well as birds, which can also be a possible vertebrate host for this virus within the sylvatic region. The primary vector is believed to be mosquitoes in the sylvatic cycle, and these have been suggested to be Aedes ceratus and Coquelidetia venezuelensis species. But really, in order for humans to come in contact with the virus, they would need to be bitten by mosquitoes in forested areas. And it's believed that the interplay between the sylvatic and urban cycles is primarily driven by human interactions. In this case, humans are essentially believed to be the ones that have brought the virus from the sylvatic cycle to the urban cycle. And it's most likely that humans are the amplifying hosts in the urban cycle. And the vectors in the urban cycle are not just mosquitoes, but also biting midges. So these would be Cuculidoides paradensis and Culex cinquefasciatus, primary vectors in the urban cycle. So you can see that there are mosquitoes in both sylvatic and urban cycles, but in the urban cycle, we also introduce midges into that process as well. There's really been no direct person-to-person transmission reported to date, but as we'll see a bit later on, vertical transmission may possibly be an exception. We know that Oropouche virus is endemic in some regions, but what's really raised the flag and sounded the alarm about concern for these viruses in 2024? There was an increase in the number of cases first noticed in Brazil between December 2023 and May 2024. And in a study of spatiotemporal epidemiology of Oropouche fever in Brazil between 2015 and 2024 that was completed by Martins Filho and colleagues, they noted that the number of cases rose sharply, specifically between December and March 2024, excuse me. The majority of cases occurred among males in the Amazon region. However, if we look at the distribution of cases across an epidemiological curve that represents the number of cases on the y-axis and the year of analysis or calendar year on the x-axis, we can see that again, over time, there are periodic bumps in positivity, likely less than 100 cases a year in this region. But again, between 2023 and 2024, the annual percentage change increased significantly to 143% when compared to previous years. So what's important to recognize that not just in 2023 and 2024, but also in previous years, this might not represent the true burden of infection because we were sort of limited with data by the surveillance systems that are in place. And it's really only in 2023 and 2024 where surveillance has really started to increase. As of epidemiological week 40, cases of Oropouche virus infection have been detected in 8 countries in Central and South America, as well as the Caribbean. The highest number of cases, and this is data that's been sourced from the Pan American Health Organization, the highest number of cases has occurred in Brazil with 8,258 cases, again, as of epidemiological week 40. The second highest number occurred in Peru. And what's most important, and we'll speak about that in 1 second, is that Cuba had also seen a significant number of cases. Now, why is it important that Cuba has seen a significant number of cases? Well, the fact of the matter is that before 2024, Oropouche virus infection was not actually detected in Cuba previously. And it's believed that this current epidemiologic, these outbreaks have spread beyond Central and South America and have reached the Caribbean as well. So this is just a map, again, from PHO that's depicting the geographic distribution and cumulative confirmed cases of local transmission of Oropouche virus within the Americas in 2024. So you can see that the primary source of infection has been the central and eastern parts of South America. And these are areas where there's dense vegetation that are prone to flooding and really where mosquitoes and midge vectors are common. The darker orange color indicates a higher number of confirmed cases. And as you can see, there are, within Cuba, there are a number of regions within the country that are now documenting confirmed cases. So it's not one specific area, it's throughout the country. And again, because of limited surveillance information, the actual case burden may be higher than what we see here. Some additional information from PHO. This is an epidemiological curve that represents the number of confirmed cases of Oropouche virus infection on the y-axis by country of identification on the x-axis. Each country is represented by different colored bars, and it follows epidemiological weeks 1 to 41 across the x-axis. Now, the 8 countries where cases have been documented are highlighted in different colors. And we see again that Brazil has the highest number of cases that have been identified, upwards of 600 cases in a week. But we also notice that the primary burden of those cases occurred within epidemiological weeks 3 and 9 in Brazil. And as time went on, we see that a number of additional countries reported infections over time. Now, some of the differences in detection here, of course, could represent, differences in rates of infection. However, it's also possible that this sort of reflects the enhanced surveillance and implementation of surveillance programs in specific regions as a result of the ongoing transmission that was identified in Brazil. And we did also see that the number of cases have started to drop off up until epidemiological week 40. However, summer in Brazil runs from December to March, so we may not have actually seen the true extent yet, and it's possible that we may see another resurgence as we get closer to the end of the year. So if we look at the distribution of travel-associated Oropouche virus infections, the primary locations where there is information on laboratory testing, either by continent or country, is primarily in 3 locations, Canada, the United States, and Europe. And these estimations are based off of available data as of November 19th, 2024. So in Canada, there have been 2 confirmed cases that have been reported by the Public Health Agency of Canada. The United States has actually seen a significant number of confirmed cases, primarily in individuals that have returned from travel to Cuba. And we'll look at a case study of returned travelers in the United States in the next couple slides. But in Europe, there's only been approximately 30 cases, with the majority of those being identified in Spain, Italy, and Germany. So again, it's possible, and again, the theme that I'm highlighting throughout this presentation is that there's limited information available, but based off of the information we do have, there's been approximately 126 travel-associated infections identified outside of the current at-risk areas. It's also possible that this number doesn't reflect the true burden of infection. This is based off of data that's currently publicly available. It's possible that some individuals may not have sought care or testing due to either being asymptomatic or not having severe symptoms. So how could you actually recognize these infections in returned travelers? And is there anything that really sets them apart from other arbovirus infections beside just travel history and ongoing transmission? The primary clinical signs and symptoms of Oropouche virus infection, um, there's really not a lot that distinguishes it from other arboviruses. The primary incubation period is estimated to be 4 to 10 days from the last time of exposure. The initial symptoms are typically mild and nonspecific, and again, that may be why some individuals may not be reflected in some of the numbers that we showed previously, because they may not have sought clinical care. Primarily, it will resemble infections and have symptoms that are similar to dengue virus, chikungunya virus, or Zika virus infections. And this is important because, again, the geographic distribution of where Oropouche virus infection occurs is similar to where we would expect these other arboviruses to also circulate. The common signs and symptoms are fever, headache, myalgia. We can have other symptoms associated with that, but it's important to note here that there may or may not be a rash. So with other arboviruses such as dengue, where a rash may be more likely to be found, Oropouche virus does not always present with a rash. What is important here is that after the first resolution of initial symptoms, so again, any combination of the symptoms mentioned above, relapse can occur. And this can occur within days to weeks after the resolution of that primary infection. And it's also possible, similar to other arboviruses, that progression to severe disease can occur, specifically neuroinvasive disease, and there have been some reports of neuroinvasive infection in several years prior in the affected regions. But typically, the neuroinvasive disease and hemorrhagic manifestations are not typical of Oropouche virus infection. So that's really the theoretical set of signs and symptoms, but is that really still relevant in 2024? Is there anything unique about current infections that might be worth investigating? So this is just a quick study that we'll look at, which was done in 2024, which looked at the characteristics of individuals where locally acquired infection was presumed in Cuba. And this was published in May of this year. So in May 2024, the National Reference Laboratory at the Institute of Pedro Curie had received samples from 89 individuals. So this was again at the early stages of infections first being identified in Cuba. And these individuals presented with an acute febrile illness of unknown etiology. Most of these individuals were noted to be dengue IgM negative. Samples that were submitted included serum, urine, and stool. And those specimens were typically collected, as indicated in the table on the left, approximately 2 days after symptom onset. And that's when we may expect viral loads to start increasing to a point where they can become detectable by our in vitro testing. These individuals primarily reported fever, headache, and joint pain for 2 to 3 days with recovery shortly after. They did not, in this set of signs and symptoms, note rash, but it wasn't clear if these individuals had a travel history or if they were really truly locally acquired infections. And it did not capture all possible severe manifestations of infections. But at least in the subset that they analyzed, the traditional set of signs and symptoms held true for locally acquired infections of Oropouche virus. In contrast, we can look at a study from the United States that was again performed earlier this year, which looked at assessing signs and symptoms among returned travelers to see again if there was any concordance or anything that was unique about the returned travelers which presented to hospital with symptoms. And this was published earlier this year by Morrison and colleagues in the Morbidity and Mortality Weekly Report. So in this study, they assessed 21 returned travelers with laboratory-confirmed Oropouche virus infection. And these travelers were primarily from Cuba, and their state of origin or residence was primarily Florida. These individuals had a median age of 48 years. 48% of these individuals were female, and most patients were assessed during the primary acute phase of the illness, or at least what was presumed to be the primary acute phase of the illness. They did note that 3 individuals had a recurrence of symptoms, which, again, as we mentioned before, differentiated individuals with other arboviral infections. Now, these individuals presented similarly as the individuals we saw with locally acquired infections in Cuba, with symptoms of self-limited febrile illness consistent with other things that have been reported in the literature. The most common reported individual symptoms included fever, which was around 95% of those individuals, myalgias, and headaches. A combination of these symptoms in combination with other um, symptoms was present in around 81% of patients. Severe illness was only noted in around 3 patients, which required hospitalization. In contrast or similar to what we saw previously, rash was noted in 6 of the 21 patients, corresponding to 29% of that patient cohort. Here, one individual also was found to have hemorrhagic manifestations with mucosal bleeding. But they did note that most patients were noted to recover without any additional sequelae, um, with the exception of those individuals that required hospitalization. So, the primary signs and symptoms are things that are behaving almost in a way that we would expect. And this is something that could be part of the differential diagnosis for individuals that are returning or that live in affected areas. However, there were also some other important clinical features that were noted, and a possibility of vertical transmission occurring in these recent outbreaks. So the Pan American Health Organization issued an epidemiological alert over infections that had been associated with adverse pregnancy outcomes. And in these cases, they noted that there were fetal deaths and congenital abnormalities that had been documented in Brazil. And in some of these cases, they noted that viral DNA or viral RNA, excuse me, was detected in fetal tissues by PCR. These tissues included brain, liver, placenta, and umbilical tissues. Some pathological findings were also noted, but at this point, there's really a very limited understanding of whether or not there is truly a causal link there between the detection of Oropouche virus and some of these pregnancy outcomes and vertical transmission. Really, the frequency of vertical transmission and whether or not timing of Oropouche virus disease during pregnancy is of risk for adverse outcomes is really unknown. It's been suggested that perhaps there is vertical transmission that may mirror what was seen with Zika virus infections. However, it's important for me to note that there's insufficient information at this time to really be able to comment further on those items. So again, this virus has been highlighted in news. We've been learning a little bit more about it as we've gone forward in today's presentation. But what are some of the factors that are contributing to this increased number of cases? And have there been any drivers that have been identified that may be changing dynamics within the Americas? There have been a number of proposed drivers of change, and these can include both environmental, biological, and behavioral factors. But there's really no clear-cut single answer to this question. Climate change is presumed to be a significant driver of Oropouche virus dynamics in the Americas. The warm and humid climates, dense vegetation, frequent rainfall, and relatively high temperatures really provide ideal conditions for the proliferation of their vectors. We also have seen that expansion of human activities, including things like deforestation and urbanization, can modify vectors' natural habitats, really increasing the risk of humans to come in contact with them, which can in turn create new urban hotspots of transmission, and can blur some of those lines between the sylvatic and urban cycles. We've also noted that human mobility and changes in human behavior might actually be linked to helping to extend the geographic distribution of vector habitats. But what's potentially the most important piece here is that there's possible virus reassortments or the emergence of new viral variants that may be driving some of these changes in virus dynamics, which we've seen in the past year. So this was investigated in a study that was published very recently by Naveka and colleagues, and they took a look at trying to look at the genetic diversity amongst Oropouche viruses that were identified in infected individuals in the current outbreak. They generated 75 genomes from Brazilian outbreak blood samples using an amplicon-based whole genome sequencing approach. And in their analysis, they noted that there was some discordance between different segments, which if you remember back to earlier, we noted that there's some regions of the genome that are more likely to be prone to selective pressures and reassortment. They noted that this had occurred multiple times between 2015 and 2024. What's important here is they noted that the current clade that's associated with the ongoing outbreak, which is referred to as BR 2015-2023, is a reassortment virus that has acquired the M segment from Oropouche BR 2009, 2018, and it has other L segments and S segments from a clade which has circulated since the late 2000s. So it has the same L and S segments as other Oropouche viruses, but there's likely been a reassortment event, which has generated a new virus variant that may be causing some of what we see in current day. How has this current outbreak affected the response in certain areas? It's really helped to help investigate and identify possible transmission cycles. It's helped to increase infection surveillance, particularly for dengue infection and Oropouche infection, through the monitoring of individuals with acute febrile illnesses. It's helped with implementation of vector control measures, but importantly also a greater recognition that preparedness, communication, and surveillance are really critical in preventing and limiting any further outbreaks of Oropouche infections. So what might be the most interesting piece and might be most relevant to everyone on the call today is laboratory testing for Oropouche virus. And I wish I could tell you lots of nice things and that there is lots of testing that's available, but the truth of the matter is, because this virus is fairly rare still, limited testing is available in Canada. It is available through the National Microbiology Laboratory, or the NML. However, it's not considered to be a routine test, and there's specific restricted indications for testing. Because of the uncertain clinical utility, it's considered to be a last resort test, which, depending on what the outcome of that result is, may or may not indicate that infection is a possibility. Both PCR and serology is available, but again, I want to highlight there is limited testing that is available. PCR is primarily performed for the S and M genes, and that's for investigational use only. Serology is available. However, there is no IgM and IgG testing that is available. It is primarily done through plaque reduction neutralization testing, and that is available through the NML at the Centers for Disease Control in the United States. However, there are specific indications for testing, and both an acute and convalescent serum must be received prior to that testing being approved. We really don't know much as well on what are the optimal specimen types and the time from symptom onset to positivity. There was a study of positivity amongst Colombian individuals that had been tested for acute febrile illness. And this figure depicts the likelihood of positivity with common Oropouche virus tests, including both PCR, which we see is the test that becomes positive the quickest after the onset of symptoms, and around 93% of individuals that were assayed were detected within 5 to 7 days. And IgM and IgG antibodies can occur further on after symptom onset. So how do you find information about testing that's available through Public Health Ontario's laboratory? We do have a test information sheet that's available on the website. we do highlight that PCR and serology are available for investigational use only. It is considered a special request and requires Public Health Ontario microbiologist approval. If a request is eligible, it will be referred to the National Microbiology Laboratory. But we do encourage that a clinical risk assessment is performed prior to requesting testing, including assessing the likelihood of alternative diagnoses, including dengue virus infection. So this is just a screenshot of what our testing information looks like on our website. We do provide detailed information for indications, specimen collection and handling, and what requisitions are required to be submitted. So similar to other arboviruses, we do require both the general test requisition as well as the arbovirus non-Zika testing intake form, and that's required for any request for Oropouche virus testing. Our current testing criteria has been developed in collaboration with the National Microbiology Laboratory and is based on guidance that has been provided by the CDC, the WHO, and PHO. Individuals that may be considered for Oropouche virus testing include returned travelers or individuals who reside in areas of risk that meet the following criteria. They have compatible symptoms. They've had travel to an area with documented or suspected Oropouche virus transmission. Testing for other relevant arboviruses has been performed. This would include testing for dengue virus, chikungunya virus, and/or Zika virus as appropriate, depending on where the individual has traveled and what the current situation is in that area of interest. As well, individuals where Oropouche virus testing may impact patient management would be part of that process. For test requests, individuals can contact Public Health Ontario's customer service. Again, we do require that both the general requisition and the arbovirus non-Zika intake form are received. When testing requests are received, the laboratory will first review tests to see if relevant testing has been ordered. If yes, the laboratory will process specimens for that initial arbovirus screening. If not, the laboratory will not process specimens for initial screening, and the submitter will be required to request testing for other arboviruses as relevant. If that submission is not received, testing for Oropouche virus will not be performed. If the laboratory is able to perform that initial arbovirus screening, so the PHO microbiologist will review that request based off of the compatible symptoms, relevant travel history, as well as negative screening for other arboviruses. If these three conditions are met, Oropouche virus requests will be referred to the National Microbiology Laboratory. If not, or unless there is strong criteria to suggest otherwise, testing for Oropouche virus infection will not be performed. So please keep that in mind when you are completing your general test requisition and arbovirus intake form. We do require information on symptoms, travel history, and screening, or at least this screening would come through our own laboratory testing, but at least compatible symptoms and relevant travel history must be provided. So finally, what is there, or what risk is there rather, to Canadians and Ontarians? The Public Health Agency of Canada earlier this year released a rapid risk assessment on Oropouche virus with specific emphasis on their implications for public health. In their assessment, they indicated that these infections were anticipated to be low risk to the general population. The greatest risk would really be to individuals that either will be or have traveled to countries with ongoing transmission. And they advised that international travelers to areas of ongoing transmission should take precautions, specifically if pregnant, given the risk of potential vertical transmission. The other pieces to note are that the climate in Canada is unable to sustain the probable vectors for cases to really be domestically acquired. So there is a travel component that will be required to this. But of course, as we saw earlier, due to the limited evidence that is available to date, risk assessments and testing approaches are not really well described for pregnant travelers. And FAC estimates that the impact to those travelers may be major. However, again, with that caveat, As more information becomes available, current recommendations and risk assessments may be updated. So really quickly, as we finish up today's presentation, one of the best methods to avoid infection by Oropouche virus is via infection prevention. I will direct you to the Canadian Ministry of Travel and Tourism, as well as CatMat, for a series of measures that have been outlined in guidance for preventing arthropod bites. These preventative measures are sort of universal for any arbovirus. This includes things like pre-travel consultations with healthcare providers, use of an insect repellent, and making sure to regularly reapply it, to limit outdoor activities during active vector times, to wear light-colored clothing that limits the amount of exposed skin, and to use things like mosquito netting and insecticide-treated clothing to, again, limit the likelihood of encountering those vectors on your travels. So, of course, there are, as I've sort of mentioned throughout today's talk, there's a lot of unknowns still regarding Oropouche virus infection. Some areas that require additional investigation include epidemiologic trends, so to better define the reservoirs, vector ecology, and transmission cycles. We really need additional information on clinical presentation, the possibility of sequelae, morbidity and mortality, as well as vertical transmission. And along with that, to really identify risk factors for severe disease. And finally, we really need to improve laboratory diagnostics, as well as the development of treatments and vaccines in the near future to help with infection prevention and control. So that concludes my presentation for today. I'd like to thank you all again for participating in the polls and being engaged, and happy to take any questions that you may have in the last few minutes that we have of today's talk.
Thank you so much, Dr. Sean Clark, for your informative and insightful presentation. We'll now begin the Q&A segment to address questions from our live audience. So to start off with our questions, in a situation where a PCR requires approval for Oropouche virus testing, is there any situations where if there is a positive result for another arbovirus, would that testing be performed at the NML, and whether or not co-infection with Oropouche and arboviruses are likely or unlikely?
Yes, sure. So I'll answer the second question, part of that question first. I think we don't know enough at this point to say whether or not co-infections are things that are possible. Of course, it is certainly possible that given the geographic distribution of the vectors and the locations where those viruses are known to circulate, it's certainly possible that that is there could happen. But right now, the testing indications, as was rightly mentioned, suggest that we would primarily be looking at individuals who are negative for other arboviruses. Right now, that information is based off of the best information that we have available from previous literature. Again, though, that's been based off of recommendations from the Pan American Health Association, Pan American Health Organization, FAC in the NML, the CDC, as well as the WHO. Part of that is because testing is not widely available. So it's really not performed routine and on special request. So it's something that requires some additional output and investment in order to actually do that testing.
Thank you for your answer. Another question that we received in the Q&A pod is that earlier it was shown in the presentation that there are no travel-associated cases yet in Ontario. Do you happen to know how many samples have been processed or submitted for testing, though, at PHO?
Yes, so we have had a number of requests for testing to date. It is less than 50.
Thank you so much for that answer. And then we have another question. Is there any discussion or consideration at Public Health Ontario to add on PCR testing for Oropouche virus to the dengue, Zika, and chikungunya multiplex PCR test that is currently available?
Yes, that's a great question. One of the challenges with onboarding testing for any of these sort of novel viruses or emerging viruses that we don't typically see in Ontario is the availability of control materials. So there is control material that is available for Oropouche virus to be able to perform these tests. And we are currently investigating implementing Oropouche virus PCR in-house. So please stay tuned in the coming months for some additional information that may follow.
Great. Thank you so much for that answer. We have another question in the Q&A pod. Are urban cycles presumed to start with human introduction, i.e. human infection in jungle, forest, or wetland environments, and then being bitten by vectors in urban settings? If so, would isolation or other precautions be recommended for confirmed cases to reduce the risk of infecting vectors in these urban settings?
That's a good question. There's with Oropouche virus, there's really, again, not a lot that's really well described. So we have limited information that's available. It is anticipated that rather than it starting with human introduction, it is, oh, sorry, urban cycles. Yes. Sorry, I'm getting ahead of myself there. Urban cycles are believed to start with human introduction. So it is anticipated that human exposure to that sylvatic cycle is really what that direct link is between those two. However, I think it's certainly possible that there may be also some distribution or changes in distribution of the vectors, and some of those lines might be blurred a little bit, particularly with things like climate change. So I think some of those cycles still need additional information to be added to them and better described for us to be able to fully answer that question.
Thank you for your wonderful answer, Sean. And as we round out the hour and conclude today's PHO microbiology rounds, I'd also like to thank you for your wonderful presentation and extend my appreciation to everyone who joined us for today's session. A reminder to everyone to save the date. Topic, the Ontario Public Health Convention will be held on March 26th and April 2nd in 2025. Check out www.tophc.ca for details. Lastly, you will receive a brief anonymous survey for today's session. Please take a moment to complete it as your feedback is very important and will help us improve our programming. Thank you and have a wonderful day.